Okay, so let's look at these membrane proteins. Membrane protein we can classify into two very big groups, which are integral and peripheral. Okay, integral, peripheral. So integral means that part of the membrane's molecule, okay, outside the membrane proteins, okay, part of them actually embedded in the phospholipids bilayer. So it means that they must have a certain part of the protein which is hydrophobic. Okay, so what does this mean? Now let us look at this. Okay, this is the phospholipid molecule. So if I have this membrane protein, let's say transmembrane protein, okay, means that they part of it embedded, right? You can see that part of it embedded, and this is a phospholipid molecule. So this kind of protein is known as the integral protein. Okay, also sometimes we also known as intrinsic protein. Okay, intrinsic protein. And we do have another kind of protein which are not, they do not embed themselves in the phospholipid bilayer. For example, you may have the purple protein, something like this. Okay, so the green protein here is known as the integral membrane protein. Okay, or intrinsic proteins. Okay, either integral proteins or intrinsic proteins. And the ones, okay, the one that does not embed themselves in the phospholipid bilayer is known as a peripheral protein. Okay, so integral versus peripheral. Now, for them to stay, always remember, for something to stay, they must have certain interaction. Correct or not? You stay with your family, you must have certain interaction, right? So for the membrane protein to stay at the membrane, they must have some interactions. So let us look at what kind of interactions. Okay, zooming in here, then let us look at this integral protein. If you look at this integral protein, I'm going to highlight two parts. The first part is here, using a gray color. I'm using the gray color. So this part of the protein must be hydrophilic. Why hydrophilic? So that they can have hydrophilic interaction with the heads of the phospholipids, or hydrophilic interaction, hydrophilic interaction with the heads of the phospholipids, as well as the aqueous surrounding. Correct or not? Okay. Then I highlight with a different color, orange color. This part, orange color, they must be hydrophilic. Hydrophobics. Okay, my mistake. Now, why hydrophobics here? Because this part of the regions, they have to interact with the phospholipid tail or hydrocarbon tail, which is hydrophobics. So what kind of interaction? Hydrophobic interactions. What kind of interaction here? Hydrophilic interaction. So hydrophilic interactions can be charged particle, positive and negative. They are charged particle, or they are polar. Basically means that one delta positive, one delta negative, forming the hydrogen bonds. Okay, so those kind of bonds don't need to worry at this moment. The next lessons, when we talk about osmosis, we will be able to see what kind of interaction. So at this moment, you need to know, for this membrane protein to stay or to anchor themselves at the membrane, they must have a certain ability to interact with the membrane's phospholipid molecule. So for transmembrane molecule, for transmembrane, means that they must have the regions with the hydrophilics, which will have the hydrophilic interactions with the aqueous surrounding, as well as the phospholipid head. And they must have the region which is hydrophobic. So this hydrophobics group are going to interact with the hydrophobic tail of the phospholipids bilayer. So therefore, they will be able to stay at the membrane there. So how about peripheral protein? Peripheral protein, then you realize that they do not need to have the hydrophobic interaction, because they never interact with the hydrocarbon tail. So means the entire regions here at the outside, they interact with the water molecule, or interact with the aqueous surrounding, or they interact with the phospholipid head. So therefore, they only need to have one property, which is hydrophilic. Are you clear? Which is hydrophilic, so that they can interact with the phospholipid head. Or they can interact, some of them, they may interact or they may have the interaction with the protein. But no matter how, you can see that the entire protein, surface of protein, they are hydrophilic. So how the protein can have the hydrophilic and hydrophobic property, we are going to learn in the next lessons, okay? Not our next topic, biological molecule proteins. Then you will be able to see how this protein will have the hydrophilic and hydrophobic regions, okay, when they fall into precise three-dimensional shape. Okay, so the integral protein, also known as intrinsic protein, they have one or more segments. They are embedded to the these are phospholipid bilayer. They can be partially embedded or totally spanned through. Okay, so the totally spanned through ones is also known as the transmembrane protein. So most integral proteins contain the residue, which is hydrophobic side chain. Hydrophobic. They can interact with the fatty acid or fatty acyl groups of the membrane phospholipids, so anchor the proteins to the membrane. So most integral protein can span the entire phospholipid bilayer. So this protein also known as a transmembrane protein. So what's peripheral protein? Peripheral protein, also known as extrinsic protein, they do not interact, highlights here, they do not interact with the hydrophobic core of the phospholipid bilayer. So they bound to the membrane by either interact with the integral membrane protein. So this is what I say, interact. Can you see that? Or they interact with the hydrophilic polar heads or hydrophilic heads of the phospholipid bilayer. So the peripheral protein can be either localized at the cytosolic phase. So cytosolic is here. So if let's say this is a membrane, double layer, right? Okay, so this is a tissue fluid. This is cytoplasm. So cytosolic phase is here. So it means that your membrane protein peripherally. So this is we call it as the cytosolic phase, or outer surface means the exo means outside, exoplasmic phase. Can you see that? So this is cytosolic, this is exoplasmic phase. Okay. So what are the role of the membrane proteins? So first, attachments to the cytoskeleton and extracellular matrix. Now, what's the difference between extracellular fluids and extracellular matrix? Matrix basically means protein structure. Are you clear? So extracellular matrix means that is the proteins of the fluids. So sometimes we call it tissue fluid. So it means the entire structure. So extracellular matrix is the component of the extracellular fluid. Extra means outside. Okay. Cell and cell recognitions, enzymatic reactivity, enzymatic reactions, or enzymatic activity, intercellular joining, signal transductions, as well as a transport which involve the ATP, okay, which is active transport or facilitated diffusion that we have learned slightly previously. So can you guys match it? Okay, 1 minute. Stop recording. Okay, now, so first, attachments to the cytoskeletons and extracellular matrix. So means that we do learn about the cytoskeleton microfilaments, right? Can change the shape of the cells. So means that they have to attach to the membrane. So membrane proteins actually is the one allowed the interaction between the cytoskeleton as well as the extracellular matrix. So this one actually refer to this, the second diagram, right hand sides. Okay, can you see that? So write down, this is the microfilaments, this is extracellular matrix. Can you see that? So the protein actually how they interact with the cell membrane, they cannot interact with cell membrane without the membrane protein. So this membrane protein, can you see that, interact with the extracellular matrix as well as the cytoskeleton. So the answer, okay, can link it. All right. Next, cell and cell recognition. So what's mean cell and cell recognition? generally, how your immune cell know, oh, this is my own cell, oh, this is the bacteria, oh, this is the fungus, how they do know, okay, about this. So cell and cell recognitions. Okay, so first of all, you can see that the cell may have the protein to recognize the glycoprotein. For example, the immune cells. The immune cells will have, okay, for example, lymphocytes. Okay, lymphocyte will have the receptor to check on, oh, this is our own cell, so don't kill. Oh, this is not, this bacteria kill. Okay, so basically cell and cell recognitions. Okay, protein recognitions. Okay, so protein and this recognition involve the glycoprotein. So answer. Okay. Now next, third one, enzymatic reaction. So what is an enzymatic reaction? Still remember in our microvilli we have learned in the intestine, right? Or I mean small intestine and large intestine. Particularly small intestine, we do have the enzyme attached on the microvilli. There is the cell surface membrane, right? So the enzyme, the protein enzyme is there to help to digest a particular nutrients, correct? Okay, so answer enzymatic actions will be the second diagram of the left hand side. Okay, intercellular joining. This is very important. In plant cells, how the two cells that they join together, they have the middle lamella, correct? Not middle lamella. But for animal cells, how the cell and the cell and cell join together, they join together through this protein interaction. So membrane protein and membrane protein interact together. So they're going to join together. So this is the answer. Okay. Signal transduction. Signal transduction, generally, we are going to learn this one once you finish the topics. So this is basically means that some signaling molecule, they cannot enter into the cell. So they need to do a signal transduction. Like for example, postman cannot enter into your house, right? But how they give a signal? They put your letter, okay, the courier man, right? Now they are online shopping, the courier man. You don't allow them to enter your house, right? But how they send things? They put into your post box, for example. Correct? Okay, so the post box, something like a receptor. Are you clear? Allowing us to actually have the signal transduction. So this is the answer. Okay, signal transductions. We're going to learn about signal transductions when we finish this part of topics. Sorry, the next topics when I had done the protein. And the last one, we look at this, the facilitated diffusion using the ion channel or active transport using these carrier proteins, right? So for transport. Sorry. Okay, for transport. So these are the answer. Okay. Any questions so far? Now, if no questions, we have done. So checkpoint 2.1 is now open for your to do a self-checking, okay, whether you understand this topic or not. So then I now stop the recording.